Hello, everyone. Welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries in programs like this. We'd like to thank the Ann and Gordon Getty Foundation and Camilla and George Smith, whose generous support made this episode of Lunch Break Science possible. Here with us today are Leakey Foundation grantee Brenna Henn and Austin Reynolds. Brenna and Austin, thank you so much for being here with us.
Thank you so much for having us.
Okay.
Brenna is joining us from California, where she is Associate Professor of Anthropology at UC Davis. Austin is joining us from Texas, where he is an Assistant Professor of Anthropology at Baylor University. Their work takes them to Sub-Saharan Africa to work on reconstructing the course of genetic diversity in South Africa. Today, they'll be discussing genetic variation, modern human origins, and working with communities in South Africa. Just a little teaser for you. They've got some really great photos coming. Before we hear from Brenna and Austin, if you're watching us live on YouTube, Facebook, Twitter, or Leakey Foundation Live, you can post questions for Brenna and Austin at any time in the chat, and they will be answering those questions live during the episode. The earlier you get those questions in, the more likely they'll be able to answer. My first question for you is, how can genetic diversity help us understand our evolution?
So genetics has been critical, I think, in helping to understand recent human evolution. And so, this dates back basically to the sequencing of mitochondrial DNA and using the variation that was observed within modern human populations to essentially assess where the origin of our species was. And of course, as is well known, the origin of the species has been now verified to be within Africa. But then we can also use subsequent variation among contemporary populations to both estimate population size, historical and contemporary, as well as migration patterns. So this is a map we put together quite a few years ago, and with now has been updated slightly. But it argues that essentially we have an origin in Africa, potentially somewhere in southern Africa, 150,000 years ago. Then this population expanded into other regions of Africa. And of course, then there is this major event which occurs roughly 50 to 65,000 years ago when the human populations then expand out of Africa. And this was accompanied by a very severe bottleneck or a reduction in the population size. So this is sometimes referred to as a founder effect, which really reduced the genetic diversity in non-African populations as compared to African populations. And this is one of the reasons that I'm so interested in studying African populations today, is they retain loads and loads of genetic diversity, which is not present in other populations around the world.
So neither of you initially thought you would become geneticists. Can you tell us when you became interested in studying anthropology and how that transitioned into approaching your questions from a genetic perspective?
Yeah, I first learned about anthropology maybe my senior year of high school, kind of going into college. I didn't really enjoy my math or science classes in high school, but I really loved my history classes and my language classes. And I found myself really drawn to the parts of our history books that were kind of on the margins. So when we were learning about Roman history, I was interested in what the Celts were doing. And when we were learning about, American history, I was interested in what all of the Native American populations were doing, not so much what, the colonists were doing. So when I was looking for college majors, I found that anthropology, archaeology, those sorts of things were the fields of study that kind of focus on these kind of more peripheral areas, and that's where all the knowledge was. And so that's where I started. My first class though was taught by a molecular anthropologist, Rika Kasel at Indiana University. And she was the first person who made the connection for me between genetics and these kind of historic and prehistoric questions that I was interested in. So after that, I was completely hooked, and here I am now, still doing it.
Brenna, how about you?
In some ways, I'm a bit similar to Austin. I was really interested in history, I think fairly early on. But when I started college, science wasn't really on my radar. And I grew up in a pretty conservative and religious area of rural Northern California. So one of the things was that evolution really wasn't taught in my high school. And when I went to college, my parents, who both have college degrees, encouraged me to take a class in evolution. So I signed up for this class on philosophy of biology. I was learning about natural selection. I was learning about the species concept debates. And I was just completely fascinated with evolution. And then I wanted, a little bit more concrete information. So then I took an anthropology class, a paleoanthropology class by Richard Klein. And then that was pretty much it. Then I knew I was going to, specialize in anthropology. And I got into genetics basically because as part of my anthropology master's degree, you had to take human biology. And one day there was a woman lecturing about the genetics of African hunter-gatherer populations, Joanna Mountain. And I just went up to her after class and I said, that was the most amazing lecture. It's so fascinating. Can I join your lab? And I had, I basically had a philosophy degree and no scientific experience. But she let me start working in lab. And I very quickly realized I was not a wet lab scientist, but I am able to analyze data pretty well. So I gravitated towards data analysis.
Well, that's a huge portion of it. So it's good that I feel like, anyway, tell us about the project you work on together and how you both became involved in it.
Sure. So my specialty is really working on African population genetics. And in particular, I started a field site in South Africa back in 2005. For this particular project for the Leakey Foundation, though, so previously I was working in the Kalahari Desert, where there are still populations of hunter-gatherers or formerly hunter-gatherer individuals that we'll talk about later. But I was interested in expanding further south. And one day I was looking at this package of rooibos tea. So you all probably know, like, rooibos is this herbal tea which is sold. And it's only grown in the mountains of Cedarburg in South Africa. And there was a picture of a woman on the package. And I looked at her and I could tell that she had Khoisan ancestry. And so I thought, you know what, we're gonna go to the Cedarburg Mountains and we're gonna start a field site there to understand people's ancestry. And so the Leakey Foundation really was kind enough to support this project and initiate our first sample collection there.
And I guess I came on the project a little later, right after it started. I defended my dissertation in 2018, and like the day after I defended, I was on a plane to California to come work with Brenna. And so my main focus as a postdoc with her was working on this Cedarburg project. I think the things that interested me the most is during my PhD, I didn't have the opportunity to do much field work or work with populations at all directly. And so I really wanted to get experience with that. So Brenna was nice enough to give me the opportunity.
And it's a gorgeous location.
In terms of the landscape, it's probably the prettiest of the locations we work in South Africa. It's really lovely.
Yeah, it's just beautiful.
They're famous for some of their wildflower blooms that occur. This is in the Tankwa, which is just outside of the Cedarburg.
Yeah, we get really lucky because we normally go during our summer, their winter. So as the field season is ending, spring in the Cedarburg is just starting. So we always get to see the wildflowers coming up.
Oh. So why did you decide to focus on South Africa?
Yeah, so there's a couple of reasons why South Africa. The first is there are a ton of paleoanthropological and archaeological sites from early humans and hominins. So here's some rock art that you'll find all over South Africa, really. And so Khoisan groups and Khoisan descendant groups that we work with have the most genetic diversity of any human population, and they kind of represent one of the earliest divergences in the human family tree. So by studying their genetic diversity, it can hopefully tell us something about, early diversity in humans, and possibly answer questions about human origins as well. In addition to this genetic diversity, Khoisan groups speak some really diverse languages, kind of famous for their click consonants. So I think we've got a video that'll come up shortly, kind of demonstrating. Yeah, so that was a linguistic map and a language school up in the Richtersveld in northwestern South Africa. And you could kind of see from the map that there's been a lot of sampling done in the Kalahari and kind of areas like Namibia and Botswana. And so one of the things that we were interested in is sampling Khoisan descendant groups kind of further south, in the Cedarburg especially. So yeah, I mean, another thing that we were interested in, particularly me, because my dissertation work was with Indigenous American populations and kind of the genetic and cultural impacts of colonialism, was what are the colonial impacts in South Africa? So this was a Dutch colony and then a British colony, and there were hundreds of years of interaction between the indigenous Khoisan peoples and Bantu-speaking peoples and European colonists. So something that I was interested in was looking at the genetic impacts of that.
Yeah. So to follow up a little bit with what Austin was saying. So when I first started working in South Africa, the population, we selected a population to work with basically based on these linguistic maps. And there were still at that time about a dozen speakers of Khmu, which is what you heard in the video. And they were the last speakers of this language, not only this language, but basically a whole subclade of this language family. And unfortunately, many of them have since passed away because they're relatively elderly. And so when you talk to South Africans, or you talk to people in general, I guess there's not necessarily an understanding that those populations still may persist in South Africa today. They just speak Afrikaans, or they have gene flow, they have admixture from Europeans or from Bantu-speaking populations. I think there's a sense that most of those populations have been wiped out, or the hunter-gatherers didn't persist. And so this is a really open question, I think, in many of the communities across the Western and the Northern Cape, who, if you talk to them, they sometimes say we don't know who we are. They sometimes say we think we have Khoisan ancestors, but we're not sure. And then you have some communities like the Nama and the Khomani-San who still have a lot of cultural retention of this information. So I think it's really interesting and critical to sample communities broadly across South Africa to understand these patterns of genetic diversity.
What's it like to work with community members in South Africa to collect samples, and what is the importance of those communities to the project?
Right. Well, the communities are everything. Like we can't do this work without their buy-in and their generous time. So I mean, I have a really wonderful time speaking with members of the communities. So I should first say that we have a great collaboration in South Africa with a local university, Stellenbosch University. So this is my collaborator, Marlon Moeller, on the far left, and another colleague, Cedric Werley, on the far right. And then that was actually one of the RAs from the Nama community on Willem in the center. So, we work really closely with local academics as well as local community leaders. And let's see. So it can be a little bit difficult to describe how you actually set up a collaboration. We have this policy where basically we kind of roll into a town and we just start talking to people. And that could be the local social workers, it could be an NGO, it could be a traditional leader, it could be somebody affiliated with a museum. And we basically say, this is what we're interested in doing. Can you introduce us to some members of the community to talk about this possibility of understanding their genetic ancestry? And so it's a lot of chit-chatting and taking people's temperature, making sure they're comfortable with this, and then we take a break. basically. So that could be a month ahead of time. It could be a whole year ahead of time. So we don't sample immediately. We like to give communities a chance to really talk amongst themselves and, let both the positives and negatives that are participating kind of percolate to the surface. And then we come back, a few weeks later, a month later, several months later, and then we actually start the genetic sampling.
Yeah, and so I can tell you a little bit more about the sampling process itself. So normally what we like to do is kind of go from house to house. So we'll have our community guide or translator with us, and we'll go from interested household to interested household. And the first thing we do, of course, is kind of reintroduce ourselves if we haven't met these individuals before, talk about what we're there for, and then get the informed consent. And once a person agrees to be part of the study, then we start with kind of a demographic, ethnographic interview. We're asking people, what languages do you speak? Are you from around here? Where were your parents and grandparents born? Things like that. And then we get the genetic sample itself. So we're collecting saliva samples, which can be kind of funny.
Austin's collecting saliva samples.
That's right, yeah.
I haven't touched them anymore.
I'm tasked with all saliva collection. And so there's a saliva tube there. And once that's done, we also collect some kind of phenotypic data as well. So things like height and weight. We've done skin color and eye color measurements as well, just to get an idea down the road of how genetic variation can affect phenotypic variation in these populations as well. So all in all, it's maybe an hour per household. And so, so I'd say we end up collecting maybe 5 or 10 individuals per day while we're there.
It's a slow process.
Yeah, it can be a slow process. Sometimes we do it in bigger settings, like community style meetings, but that's not really our preference. It's a little rushed.
It's gotta be really neat. You probably hear some really fun stories.
We do, and this can be quite an emotional experience for certain participants because many of them, they grew up during apartheid, so they grew up during a period of time in South Africa where there was a deliberate attempt to erase these, cultural memories or the languages that people were speaking, and they were forced to live in particular areas. And there was a lot of racial segregation. And so, we have interviews where people start crying because we ask them about their family history and they describe how they weren't allowed to walk past a white person, to touch a white person. And now here we are in front of them, like taking down their oral history and giving them a hug and everything. So I think, it's an emotional experience for both us as the researchers as well as the participants. But it's also, it can be very fun as well, because you do get to hear some of these oral histories or these just like cultural stories, folk stories that people have. And so when I was working in the Kalahari, I remember talking to people about the different languages. So the new language or Nama that they spoke, these click languages, and one participant telling me, oh yes, you speak Nama, so that the animals basically they understand Nama. And he described going over a dune in the Kalahari and seeing a lion, and then speaking Nama to the lion, and then the lion goes away. That was they can communicate with animals in the traditional languages. So I have a wonderful time chatting with folks.
Austin, do you have any fun stories or?
Yeah, one comes to mind. We were doing some sampling in the Karoo, and while we were talking to some members of a household, there was a grandma who kept going out into the yard, and it looked like she was tending a fire. And I was really interested in what she was doing. It was like kind of like a circle of reeds or dry grasses, and then a fire in the middle. And apparently this is like the traditional way to cook flatbreads, which of course the name is escaping me now.
The roastabrot.
Yeah, roastabrot. And so I spent maybe 10 minutes talking to this Oma, this grandma, about like, how does she make the roast of wood? Why do you do it this way? And I got a whole lesson on how to do this and why we do it that way. So it's pretty cool.
So how do you define a population from a genetic perspective, and what does that mean to the communities you work with?
Yeah, so I think for us, we tend to define populations in kind of an ethno-linguistic manner, and we kind of let our participants lead that definition. So part of our demographic interviews are we're asking how people like to be called, what their ethnicity is. And so when we're working in places like the Richtersveld, where we have the Nama community, everybody that we talk to will say, I am Nama. But working elsewhere, like in the Cedarburg, people don't have that kind of strong cultural memory of a Khoikhoi or San population group, so they default to colored, which is kind of this ethnic category that was kind of defined by colonial and apartheid governments as kind of like a catch-all for Khoisan people and people of mixed descent. So in that case, where people they're not identifying as Komani or Nama, they're just colored, we kind of use a more geographic approach where we've got population from Cape Town or population from the Cedarburg Mountains or elsewhere. So yeah, it's really all about how our participants view themselves, really, is how we split things up.
And you have a fun video of something you've got to see while collecting. Do you want to introduce it?
That's right. Yeah. So we'll talk a little bit more about the colored ethnicity during our kind of brief talk. But whereas Brenna was saying earlier that a lot of these communities don't know about their connection to Khoisan peoples, or they've kind of forgotten that cultural memory, a lot of cultural characteristics or cultural practices still persist in these communities. So this will be a video of the real dance, which is a Khoisan dance that's still practiced by colored communities today.
It looks really hard.
No,
Did either of you try it?
Oh, no.
No, I would be terrible.
I missed it, actually. I don't know what I was doing when Dan and Austin took that video. I guess I was back at the house probably doing paperwork or something fun, and they got to see it. So, I guess to follow that up, we're not in a position to tell communities how to identify. But of course, genetic ancestry can be an important component of someone's identity. So, I think it's really important for us as well as other geneticists to make sure that you go back to the communities that were sampled and return the results. And so, this has been something that is just, I think, really, really critical for our work in South Africa. And usually we've done it in a variety of ways. So often we'll go back and, we'll try to assemble a bunch of people in a church or a soup kitchen or, whatever location can accommodate 30 people, and then we'll give a short presentation. And so sometimes that works really well. We've done community posters and then put them up in like, local tourist halls. We've also done radio interviews. So we try really hard to make sure that we can go back and speak with the community members. And I would say, by and large, the experience has been incredibly positive. Like this is what everybody wants. They want you to come back and tell them what you learned. So, for example, this is a little video we made just illustrating very recent migrations from Eastern and Western, and there'll be an image from Europe coming into Southern Africa over the last 2,000 years. And so just being able to communicate those different periods in their history, and then sort of letting people ask questions about it. Again, I think it's probably the most rewarding part of the work.
Well, I'm excited to hear more about your research. Before we turn the virtual floor over to Brenna and Austin, I want to give a shout out to everyone watching this episode of Lunch Break Science live on YouTube, Facebook, Twitter, or Leakey Foundation Live. It's exciting to see all your locations. If you are enjoying this episode of Lunch Break Science, be sure to subscribe to our YouTube channel or follow us on Facebook or Twitter to receive reminders of upcoming episodes. Now let's turn over the virtual floor to Brenna and Austin and hear more about their research.
All right, Austin, I'll let you cue up the slides. Thank you. Okay, so we're just gonna do a quick little lecture to supplement all these questions. So we've been talking about these populations throughout, and we've been throwing a variety of names at you. You're probably, as the general audience, may be familiar with terms like Bushmen or San or hunter-gatherers, right? So these often have referred to populations living in Southern Africa and practicing a particular subsistence strategy. But of course, the people that we work with today are completely contemporary individuals, many of whom have transitioned to wage labor or farming. And so, this is an example of what people look like. But sort of very interestingly, as we mentioned, many of them now self-identified using this term called colored, which is really an apartheid era term. And it's sort of unclear what ancestry that can often connect with. Next slide, please. So from a genetic perspective, these populations are, especially valuable or especially unusual because of the very, very high levels of genetic diversity that they carry. And so this is from a study that we did a few years ago, sequencing about 7 individuals from 7 different populations across the world. And you can see that in the purple here, so that's the Namibian San population, the Twasi population that's represented, and they carry the highest number of heterozygotes of any population sampled in this study. So just to remind you, if you've forgotten your high school genetics, a heterozygote is a location in your genome where you have 2 different alleles. So a C and a T, or an A and a G. And if I went through your genome and just counted up the number of locations where there's a heterozygote, in the San population, we see that there's 2.4 million on average, whereas let's say in the Cambodian population, there's only about 1.8 million. So there's a really, really large difference actually if we sequence the heterozygosity in somebody from Southern Africa versus somebody from East Asia. Next slide, please, Austin. And so we're trying to understand better right now, how did this diversity emerge historically? I think these patterns of genetic diversity are fairly well characterized now, but what is the actual process by which these patterns are formed? And so one of the, I think, more exciting things that I'm involved in with the moment is having sequenced hundreds of genomes now from several populations in Africa, including the Nama here, who are located in green. So those are a South African Khoikhoi population from the Namibian-South African border. And what you can see, sorry, if you just tab once, maybe Austin thinks, that they're the first population to basically split off from all other African populations. So that is the Nama diverge really 100, probably less than 150,000 years ago. And then West African populations like the Mende, East African populations like the Gamuz or the Amhara, they diverge at a later point in prehistory. So I think this is potentially an explanation for why we see so many unique mutations segregating in Khoisan populations, and really sort of why they look quite unusual relative to many other African groups. So this is a exciting kind of ongoing project. Next slide, please, Austin. So just to maybe back up slightly and talk a little bit more broadly about our fieldwork. In my lab, we really work basically in several locations across South Africa. So we have field sites in the Richtersveld with the Nama population, in the southern Kalahari with the Komani-San, as well as the tuberculosis cohort. And then in the Cedarburg and the Karoo, so that's the orange population here on the map, and the Karoo is just to the east of that. So that's where we're going to sort of focus on our results today. And to date, we've collected about 3,000 DNA samples from Southern Africa. And right now we have this very ambitious goal of growing this to over 10,000, partly through our tuberculosis cohort, but then also doing these smaller ethnographic based studies as well. Next slide, sorry. So I think you've seen some of these photos that we've put up that were taken by some fantastic graduate students in my lab. So, you know, sometimes it's very serendipitous. You'll be driving down the road, and for example, this is a man in the Cedarburg, and he's actually a herder. You can see his whip lying on the ground there for the sheep and the goats. And you sort of just jump out of the truck and say, hey, do you have a few minutes? And you talk to him about the study, and then go through the consent form. Next picture, please. And we always do this also with another member of the community present, so who both helps as a translator, but is also there to make sure that whatever words we're using, whatever we're asking the community member to do is sort of culturally appropriate. So we always have another witness as well. Next slide, please. Thanks. And just to emphasize that this is really done as a team. So usually it's not just me, but you know, I'll often have Austin there as my postdoc or a graduate student, my project manager, Justin Myrick, other collaborators from Stellenbosch University, such as Marlu or Cedric or Caitlin. Yeah, so it's really, really wonderful for all of us to work together. Next slide. So we're collecting a variety of different types of data, and I think Austin mentioned some of this earlier, but all the DNA is really derived from saliva. So we just give people these little tubes and ask them to spit into the tube, and it's a very small amount of saliva, and most people are very happy to contribute a fluid which they don't particularly value, so that hasn't generally been an issue. Additionally, we collect some phenotypes such as skin pigmentation, height. We're really interested in eye color as well, because there's a lot of phenotypic variation in these populations, and we've already sort of found some kind of surprising genetic results from connecting the genetic variation to these unusual phenotypes. And then I think sort of more pertinent for this lecture is we also do a demographic survey. So we ask, you know, where was your mother born? Where was your mother's mother born? Where was your father's mother born? What languages did they speak? And by doing so, we can actually collect a relatively large data set with all of this really valuable information, including languages which are no longer ever even spoken today, that are essentially extinct languages. I would say like that's happened several times. I have, I don't know, at least 3 different languages represented in our collections that are considered extinct languages today. And next slide, please. So just to sort of quickly introduce the prehistory of these populations more generally, right, as I mentioned, the Khoisan likely diverged from other African groups as early as 150,000 years ago, but they haven't been completely isolated. So we know that 2,000 to 3,000 years ago, there's likely this migration from Eastern Africa of people who were pastoralists, so people who had sheep and cattle and goats, and they moved down through the Great Lakes region and into Southern Africa. So they contributed both this herding culture, which is frequent now in like the Nama population, as well as some genetic signatures. Then about 1,500 years ago, the Bantu-speaking populations really moved in from Western and Central Africa, and particularly spreading sort of through the Kalahari Desert and then the eastern part where it's wetter in South Africa and along the Cape. And then of course, there's this major genetic impact from European arrivals, and they began spreading out from basically a point in what is now considered Cape Town into different areas of South Africa. And Austin is going to tell you much more about that part of it.
Right. Thank you, Brenna. So we've been talking about how we can utilize genetic diversity from Khoisan groups to understand something about deep human history. But to do that, we kind of have to dig down through the genetic impacts of colonial history, which has been kind of the past 300, 350 years in Southern Africa, and look at the effects there. So I'm going to briefly walk you through colonial history of Western South Africa. So the Cape Colony was first founded by the Dutch East India Company, the Cape of Good Hope, around 1652. And for the first 50 years of the colony, it was really just the Cape Peninsula. They traded for livestock with the local Khoikhoi and San populations. But around about 1700, the Dutch East India Company decided that wasn't enough. So they brought farmers in and spread around into kind of Cape Flats area, so all this flat land just north of the Cape Peninsula. And they didn't reach the Cedarburg Mountains, or the study area that we're focusing on today, until around about the 1730s. So this is where the Cedarburgs are. And the Cedarburg Mountain area was kind of this frontier region for over 100 years, where there were settlements, but there were also independently living Khoisan communities, as well as kind of escaped enslaved individuals. It was kind of like the Wild West of South Africa for a long time. And the colonists didn't spread north to the Richtersveld until the early 1800s. That's where the Nama lived today. And they didn't get out into this dry region of the Karoo until at least the 1840s. So there was a long period of time in a lot of our study regions where there wasn't a colonial influence. But today we'll focus on kind of the Cedarburg. So we'll start with the genetic results here. So at the bottom here, we have what's called an admixture plot. And for those of you who haven't seen one of these before, I'll just briefly walk through what it's saying. So this is a bar plot, essentially, where every kind of single bar or line here is representative of an individual in the study. And all the different colors represent the ancestries across their genome at a different kind of set of populations. So in this particular admixture run, we told the program to split it up into 6 different ancestral populations. So it's going to go through all of our study populations, split them up into 6 groups, and kind of paint everybody's genomes, the colors which correspond to those groups. So we've got here on the left, Bantu-speaking population, the Luya from Kenya. Moving to the right, we've got the Maasai, a Nilotic-speaking group from Kenya, Palestinians from Western Asia, British populations, Han Chinese, and then we have South Asian groups, the Bengali and the Tamils from the 1000 Genome Project. And you can see that kind of each of these populations at k = 6 are kind of split up into individual ancestry groups. And then if we go to the right, our study groups, a subset of the Komani individuals and the Nama pastoralists, you can see this large kind of red area, which is the Khoisan ancestry. And if we look at the Cedarburg specifically, we found that on average, people in the Cedarburg have about 50% of their ancestry from Khoisan ancestors. So that means that 2 of their grandparents were Khoisan. About 25% of ancestry in the Cedarburg on average is from Europeans. So that's kind of 1 grandparent's worth of ancestry. And then another 25%, the last portion of their ancestry, is made up of either Bantu-speaking African or kind of East and South Asian. So on average, people will have kind of 1 great-grandparent that was Bantu-speaking African ancestry and 1 great-grandparent that was of East or South Asian ancestry. And you can kind of see, comparing the Cedarburg to the Nama or the Komani, they have quite a bit more European and then East and South Asian ancestry. And if we look at just the Khoisan components, we actually see a strong correlation between the distance from Cape Town, kind of the major colonial and now economic center of Western South Africa, and the amount of Khoisan ancestry. So if we look at colored individuals collected in Cape Town, they have the lowest amount of Khoisan ancestry here on the y-axis, Cedarburg individuals, and then moving up to the Nama and the Komani, who have the most ancestry. So a really strong connection between the amount of Khoisan genetic ancestry and the distance from Cape Town. So this got me wondering, we see this kind of geographic pattern of Khoisan ancestry. Do we also see a geographic pattern of cultural traits as well? So that is something that we did. So we used this demographic information where we're collecting the birthplaces of people's parents and grandparents to reconstruct what were the migration patterns like over the past 3 generations, or the past 150 years, give or take. And so using this data, we can learn something about what's called post-marital residence patterns. So where a couple chooses to live after they are married. So every culture will have a preference for some post-marital residence patterns. So some groups are matrilocal, where they tend to live with the wife's family members after marriage. Some groups are patrilocal, where they tend to live with the husband's family afterwards. And that's just really simply speaking. Although, even though a population might have an express preference for one of these, we know that not everybody always fits into these patterns. But in the Cedarburg, what little ethnographic and historic data we have suggests that the Khoisan populations there may have been practicing a matrilocal post-marital residence pattern. And this gives us some predictions of how people should be moving. So under matrilocality, men should be moving more often than women because they're moving to live with their wife's family when they get married. And men should also be moving further distances than women as well. So we use these kind of longitudinal data set of people's birthplaces to look at that in the Cedarburg. So here we have kind of the proportion of people who migrated on the left, and then their median distance of migration on the right. And we can see that in the Cedarburg, in the grandparental generation, men are indeed moving more often than women, and they're also moving further distances. But in the parental generation, these are the parents of our participants. Men and women are moving at roughly equal rates. This could be a kind of function of modernization, where people are no longer adhering to these kind of traditional post-marital residence patterns, but men are still moving further distances than women. And when we compare what we have in the Cedarburg to the Richtersveld, which is kind of twice the distance away from Cape Town as the Cedarburg is, we see similar patterns where men are moving more often than women, and they're also moving further distances. And we even see that during the most recent generation in the Nama and the Richtersveld, men are still moving more often than women. So what we think we're seeing here is not only kind of a geographic gradient in Khoisan genetic ancestry, but also possibly a cultural gradient as well, where some of these Khoisan cultural practices are persisting in populations further away from the colonial and economic center of Cape Town. So I guess just to wrap up, first and foremost, we want to thank all of our participants. Without them, obviously none of this work would be possible. In addition to the Leakey Foundation for funding our sample collection in the Cedarburg Mountains. And Brenna, who else would you like to thank?
Just everybody on the team as well for coming out and collecting.
That was a fascinating talk. I'm really excited to get into questions. We will be now taking questions from the audience. If you have a question for Brenna and Austin and have not gotten your question in, please be sure to get it in right now. So let's take our first question from the audience. Let's see here. It comes from Lou. Hi, Lou. What other phenotypic data do you collect other than height and weight, if any?
Great question. So yeah, right now we're focused on skin pigmentation, eye color, and height in most of these collections that we've been talking about today, the Nama, the Kumani-san, and then the Cedarburg and the Karoo colored populations. However, we are doing much more extensive phenotyping for this tuberculosis cohort, which is really sort of centered in Uppington and then rural health clinics around Uppington. And so in those cases, we're collecting people's status in terms of tuberculosis, HIV, body mass index, so height and weight, hip and waist weight ratios, what sort of medications they were on to treat tuberculosis. So we have a whole phenotype survey there, but and that study really, is much more clinical because the goal is to try to understand what are the genetic determinants of tuberculosis activation in the colored populations in South Africa, and is that unique to the variation that's segregating there, or is it also genetic variation that contributes to TB susceptibility in other groups as well? But thanks for your question.
Our next question comes from H. Gregory. How have you continued your research and work during the pandemic?
Austin, you want to take that one?
Sure. Yeah, it's been difficult. I think for the first year of the pandemic, we didn't do any field collection. We had planned to go back in the summer of 2020, particularly for the tuberculosis study, but also to do some return of results. And that wasn't possible. This past spring, as vaccines started to get rolled out, and South Africa went down to kind of a lower level of alert, if you like, in terms of movement around for the pandemic. Our field manager, Justin, was able to go back, but only for a few weeks, because he was just there for a few weeks and had started getting samples collected again, and then COVID cases started to be back on the rise in South Africa, so we couldn't collect samples anymore, so we had to go. But we're working on getting everybody vaccinated on our research team, and we're hopeful that next year, kind of the global response and vaccine availability will be a lot better, and so everybody can be vaccinated, so we can resume normal operations, if you like.
And I think it's just important to add that any interaction that we have with participants is actually governed by human subjects at Stellenbosch University. So, we have to apply for permission basically to be there in South Africa and to be working in these communities or talking to people. So yeah, we don't do any of that without official permission from the Human Subjects Board.
Our next question comes from Oshioma. She asks, what is the yellow ancestry in the admixture plot? And I think we have the plot. I think this is Is this the correct one?
Yeah. So that's I'd call it like a Western Asian component that we see in both the Palestinians and the Maasai from Kenya. And this is kind of representative of spread of pastoralism from Western Asia into Eastern Africa. And then we include it because we think there was also a spread of these pastoralists down into Southern Africa as well. And it would have brought kind of the first sheep to the region. So you can kind of see on the right in the Nama and the Cedarburg especially, there are kind of small components of that yellow ancestry. Yeah. Anything you want to add to that, Brenna?
No, that's good.
There you go, Oshi.
Okay, our next question comes from Jeannie. What were any Khoisan people enslaved and sent to the Americas after the Dutch and English arrived?
Ooh, great question. Oh, well, we'll both try to answer this. So my understanding is that a lot of the slave trade basically it extended as far south as Angola, and there are Khoisan populations in southern Angola, but I don't think that they were sort of frequently enslaved. There was also slave trade on the eastern part of South Africa. So, but I don't think there's that much that shows up in like African American or Afro-Caribbean populations. I think I've informally spoken to someone at 23andMe and asked whether or not they detect Khoisan ancestry in participants in their database, and they said it's fairly infrequent. But I don't know if anybody's ever tried to really quantify this. I don't know, Austin, maybe you have more information.
Yeah, I haven't seen anything published about Khoisan ancestry in African American or Afro-Caribbean populations. But I'll just add to what Brenna was saying in that kind of South Africa and everything Cape Town East was involved in the Indian Ocean slave trade and not the transatlantic slave trade, which we're more familiar with in the States. So a lot of the enslaved individuals being moved in the Indian slave trade were from Madagascar or the Indonesian archipelago, places like Malaysia, as well as South Asia as well. Sri Lanka was a major source. So a lot of the kind of ancestry that we see in admixed populations in South Africa today from previously enslaved communities are going to be those Eastern and Southern Asian components.
We have another question from Lou. He asks, is it more common here than, I think, to spell Khoisan with an I rather than E? Is it more appropriate to use an E?
Oh boy, there's so much nomenclature around the terms and how to spell them. So I've sort of defaulted to using the I, so Khoisan, only when I'm talking about the language families. But then when I talk about the populations, I tend to use the E, and also I tend to hyphenate the Khoi, so it's Khoi-San. And this is partly a request from the communities that while they recognize that the ancestry of the Khoi and the San are similar, they're culturally distinct communities. And so they would like to be represented as distinct communities. And having the hyphen in there kind of lets people know that it's two different groups that are sort of being lumped together rather than just sort of one single group. So yeah, it's, the literature on this is, I remember when I started as a graduate student trying to understand all the different population labels and colonial labels and post-colonial labels. And my God, it's crazy. But yeah, so I stick with the E when referring to populations.
Our next question comes from Brandon. Brandon asks, what is the biggest challenge when trying to recruit people to participate in your research?
Go ahead, Austin.
I'm trying to. I will say that by and large, everybody that I've had a chance to talk to is really excited about our research. So when we just show up and start asking questions about history, my general response has been one of kind of surprise, and they want to know, like, wow, why are you asking us about this? So once we explain why we're there, people are generally really excited to learn more about their history through genetics, and to just have us there asking questions, it seems. Some of the biggest challenges can be really just getting around to everyone. Our most recent field season, we had a lot of flat tires. I will say we weren't using our normal field vehicle, which is very rugged. We were using that's our normal field vehicle. No problems there. We were using a rental vehicle, and we fixed no fewer than 4 flat tires. So yeah, getting around can be difficult. Brenna, maybe you can speak a little better to the challenges.
I mean, I think from a participant standpoint, I would say, 9 out of 10 people say that they do want to participate in the study. We have a very positive, robust response to the type of projects that we're doing. Occasionally, I've had interactions where actually maybe one of the younger members of the household is opposed to her parents participating in the study. And I think, a lot of this has to do with racial tensions in South Africa and the way that people were treated during apartheid. But very interestingly, like, the older generations actually do want to tell a lot of their story, and they sometimes say that, like, the younger generation doesn't want to hear it. They don't want to have to sort of relive the painful memories that their grandparents went through. And so, yeah, I would say sometimes there is a sort of a generational tension about participating in this. But by and large, yeah, most people seem very interested.
My last question for you both is, do you have any advice for those who are watching who are interested in pursuing and studying anthropology?
Yeah, I mean, I think saw that both of us didn't start out college kind of thinking that genetics or anthropology was even going to be what we ended up doing long term. And genetics can be a really technical field. There's a lot of math and really computer programming now that's involved. And so I would just say that anybody who's interested in this sort of thing, I think that you can teach yourself anything if you're interested. What really spurred me to kind of relearn calculus and get going with computer programming was my interest in human genetics and what learning those tools could help me with when I was studying human genetics.
I second Austin's response and take an anthropology class and
Yeah.
see if this turns you on, and then it's highly motivating, as Austin mentioned.
Well, thank you again so much for joining us, Brenna and Austin. I thought it should be fun to end the episode with this great photo of you.
Relaxing after a long day.
Yeah.
Looking out on the sheep field. There.
So I am thrilled to announce that Lunch Break Science has been renewed for more episodes, and we will be having no new episodes in September, the month of September. As Lunch Break Science team and I are preparing for new episodes starting Thursday, October 7th. I will miss you all in September, but be sure to check out these upcoming Leakey Foundation programs. Next time on Lunch Break Science, we meet primatologist Alba Garcia de la Chica and learn about the mechanics that allow maintenance of pair bonding and monogamy in owl monkeys. It's going to be a really good program. The final program in our exclusive summer travel series is on August 26th at 5 p.m. Pacific. You'll be virtually transported on a whirlwind tour of several sites that have received funding through the Primate Research Fund. Dr. Takeshi Fukurishi takes us to the forests of the Democratic Republic of the Congo, where the first ever ecological and behavioral study of bonobos was established. Dr. Jill Preetz takes us to a mosaic environment in Senegal, where we'll see how dry savanna environments influence chimpanzee behavior. And lastly, Dr. Caroline Shupley takes us to the forests of Sumatra in Indonesia, where the Suak orangutans are exhibiting complex behaviors such as tool use. Attend this program live for a $100 donation or receive a link to the program recording for a $10 donation. All donations to the Primate Research Fund will be quadruple matched thanks to generous sponsors. You'll protect vulnerable primates, long-term field research sites, and their local staff members, have an exclusive look behind the scenes at some pretty amazing locations, and much more. We've shared the link to this program in the chat. We hope you will be able to join us for these, wonderful programs. Thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until October, stay hungry for knowledge.
Thank you.
Lunch Break Science is brought to you by the Leakey Foundation and made possible by the generous support of the Anna Gordon Getty Foundation and Camilla and George Smith. Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, today's guest scientists, and how you can help support human evolution research and educational programs like Lunch Break Science. Right now, all donations to the Primate Research Fund will be quadruple matched by generous donors, meaning your impact will be quadrupled. Miss an episode of Lunch Break Science? Catch up on past episodes and browse our library of Leakey Foundation lectures on our YouTube channel. Still hungry for science and can't wait till our next episode? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Subscribe to Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about exciting upcoming episodes and programs, as well as groundbreaking discoveries in human evolution research.